Deep underground, in rock layers that are 65 million years old, scientists have found something unexpected. Tiny metal spheres, made of rare elements, are scattered across the planet. Their pattern looks almost identical to the fallout left behind by modern nuclear explosions. But these spheres were formed long before humans ever walked the Earth. We've always believed that we were the first to split the atom, to shape metal, to burn fuels, and to change our planet's air and climate. But what if that belief is wrong? What if advanced civilizations once existed here, long before us, and time has simply erased every trace of them? Leave a like and subscribe to the channel, as in this journey, we'll look at clues that question everything we thought we knew about Earth's past. We'll explore strange chemical signatures that seem to hint at ancient nuclear events, study the great die-offs that reshaped life, and ask why the fossil record holds such huge, puzzling gaps. Consider attempting to construct a functional smartphone by randomly shaking electronic components in a container for 600 million years. Life's emergence on our planet defies every statistical expectation. Once our world cooled sufficiently to maintain liquid water, something remarkable occurred within what scientists regard as a cosmic instant. Intricate molecules began self-organizing into replicating systems. These weren't merely simple carbon-hydrogen chains, but complex biological engines capable of precise self-duplication. Earth formed roughly 4.5 billion years ago, spending its first 800 million years as essentially a massive lava lamp under constant asteroid bombardment. Surface temperatures soared to 2,000 degrees Fahrenheit, making survival impossible. Yet by 3.8 billion years ago, evidence of sophisticated microbial life already appears in the fossil record. This provides evolution merely 600 million years to progress from sterile rock to functioning organisms. Charles Darwin himself identified this rapid emergence as the warm little pond problem, recognizing that natural selection demands extensive timeframes to generate even rudimentary life forms. The complexity involved is staggering. Basic bacteria contain approximately 4 million individual components functioning in perfect coordination: DNA, RNA, proteins, cell membranes, and energy-producing systems. It resembles discovering a fully operational Boeing 747 in a scrapyard that contained only raw materials the previous day. Contemporary scientists have attempted recreating these conditions in laboratory settings, mixing appropriate chemicals and applying electrical discharges to simulate lightning. The results? A few basic amino acids, protein building blocks. Nothing approaching a living cell. The chasm between chemistry and biology remains as enigmatic today as during Darwin's era. An alternative explanation makes this timeline comprehensible. Life didn't originate from scratch on Earth. It arrived already assembled, already tested, already functional. Envision life as a cosmic inheritance transmitted from civilizations that solved biological puzzles billions of years before our solar system's existence. This transcends science fiction. NASA has discovered organic compounds throughout the galaxy. The European Space Agency has identified complex molecules in interstellar clouds. Life's building blocks aren't rare cosmic flukes. They're ubiquitous throughout space. The Panspermia Hypothesis proposes that life distributes throughout the universe like wind-borne seeds. Some seeds land on appropriate planets and flourish. Others drift through space for millions of years awaiting suitable conditions. From this viewpoint, Earth wasn't life's birthplace. It was simply another garden where cosmic seeds finally discovered fertile soil. Every day, Earth receives 100 tons of space debris. Most cosmic visitors incinerate in our atmosphere, creating shooting stars. But some survive, crashing as meteorites. For over a century, scientists cracking open these space rocks expected minerals and metals. Instead, they've uncovered something far more extraordinary. The Murchison meteorite revolutionized everything. When this 4.5-billion-year-old rock struck Australia in 1969, researchers discovered something impossible inside: amino acids. Not just any amino acids, but 74 different varieties, including many absent from natural Earth chemistry. These weren't contamination from our planet. Chemical signatures confirmed their deep space origins. Amino acids function as biological building blocks. Every protein in your body, from oxygen-carrying hemoglobin to food-digesting enzymes, is constructed from these essential components. Discovering them in meteorites was like finding a construction kit for life floating through cosmos. The Murchison meteorite wasn't unique. The Allende meteorite from Mexico contained even more sophisticated organic compounds, including nucleotide bases, the genetic code letters in DNA and RNA. The Murray meteorite revealed sugar compounds forming genetic material backbones. The carbonaceous Hill meteorite contained lipids, cell membrane building blocks. Some meteorites predate our solar system, formed in stellar nurseries billions of years before Earth existed, transporting their biological cargo across unimaginable distances and time spans. It resembles receiving bottled messages from civilizations that existed and perished before our sun's birth. Recent analysis has revealed something more shocking. Some meteorites contain microscopic structures appearing exactly like fossilized bacteria. These aren't random mineral formations. They possess distinct shapes, sizes, and internal organizations of living cells. The ALH 84001 meteorite from Mars sparked intense debate when NASA scientists claimed evidence of ancient Martian life preserved within its rocky matrix. If meteorites regularly transported organic compounds throughout the early solar system, then life's building blocks were being delivered to planets like a cosmic delivery service. This delivery system remains active today. Every meteor shower represents potential life seeds raining onto our planet. Earth occupies the most perfect position imaginable, 93 million miles from an ideally sized star. We call this the Goldilocks zone. Not too hot, not too cold, but precisely right for liquid water. Move us 5% closer to the sun and we become Venus with lead-melting surface temperatures. Shift us 10% farther and we transform into Mars, a frozen desert where water exists only as ice and vapor. The mathematical precision is breathtaking. Our planetary orbit varies less than 2% annually. Our moon is exactly the right size to stabilize our axial tilt, preventing wild climate fluctuations that would make complex life impossible. Our atmosphere contains 78% nitrogen and 21% oxygen, a combination so chemically reactive it should have burned away billions of years ago. The odds of this occurring by chance are so minuscule that scientists struggle expressing them mathematically. It resembles flipping a coin and obtaining heads 10,000 consecutive times. But Earth wasn't always this perfect. For billion years ago, our sun was 25% dimmer. By all calculations, our planet should have been a frozen snowball, completely inhospitable to life, the faint young sun paradox. Yet during this supposedly impossible period, life not only existed but thrived. Geological records show clear evidence of liquid oceans and complex microbial ecosystems. Something maintained Earth's warmth when it should have been cold, sustaining conditions allowing life to establish and spread. The Panspermia Hypothesis offers a different perspective. What if Earth's perfect conditions aren't accidents but engineering? What if advanced civilizations, facing their world's deaths, didn't randomly send genetic material? What if they deliberately selected target planets and modified them for life? Consider the required precision. You'd need to adjust atmospheric composition, orbital mechanics, magnetic field strength, and planetary rotation. You'd need to account for stellar evolution, ensuring your chosen world remained habitable as its star aged and brightened. This terraforming level would require knowledge and technology far exceeding our current capabilities. Deep in the Atacama Desert, where it hasn't rained for over 400 years, scientists have discovered bacteria thriving inside rocks. These organisms, called endoliths, live in conditions that would kill almost any other Earth life form. No water, no oxygen, no organic nutrients, and lethal radiation levels. Yet they not only survive but reproduce, creating entire ecosystems within apparently solid stone. This discovery prompted a startling question. If life exists in Earth's most extreme environments, what about the ultimate extreme environment, outer space itself? The answer emerged from International Space Station experiments. Scientists exposed bacteria samples to space vacuum where temperatures swing from 250 degrees below zero to 250 degrees above. With no air pressure, no radiation protection, no nutrients, no water, conditions that would kill humans instantly. The bacteria not only survived but continued reproducing. Deinococcus radiodurans, nicknamed Conan the bacterium, withstands radiation doses 3,000 times higher than lethal human levels. It survives complete vacuum for years. When cosmic rays shred its DNA, it simply rebuilds itself using internal repair mechanisms scientists still struggle understanding. Tardigrades, microscopic animals resembling eight-legged bears, are even more impressive. They can completely shut down their metabolism, entering cryptobiosis, a state neither alive nor dead. In this state, they survive temperatures of minus 458 degrees Fahrenheit, just degrees above absolute zero. They handle pressure 6 times greater than the deepest ocean trenches. They can go without food or water for over 30 years. When researchers exposed tardigrades to space vacuum for 10 days, 70% revived once returned to normal conditions. They resumed eating, reproducing, and behaving as if nothing happened. These aren't isolated examples. Scientists have found bacteria living in nuclear reactors, concentrated acid pools, boiling hot springs, and Antarctic ice frozen for millions of years. If organisms this resilient exist naturally on Earth, imagine what life forms might exist on planets with harsher conditions, worlds orbiting dying stars, planets with thick toxic atmospheres, radiation-bombarded ice moons. Life might not just survive in these environments, it might thrive. This revolutionizes interstellar travel understanding for living organisms. We once thought life was fragile, requiring perfect conditions for even short space journeys. Now we know some Earth organisms could potentially survive trips lasting thousands or even millions of years. If bacteria and tardigrades can survive in space, they could be hitchhiking on asteroids and comets right now, traveling between star systems like biological stowaways. Charles Darwin called it the most serious objection to his evolution theory. The fossil record, which should display gradual transitions between species, instead reveals something far more puzzling. Complex life forms appear suddenly, fully formed, without clear ancestral lineages. It resembles finding modern smartphones in archaeological layers without evidence of earlier communication devices. Consider the Cambrian explosion 540 million years ago. For 3 billion years, Earth was populated almost exclusively by simple single-celled organisms and basic multicellular forms. Then, in a geological instant spanning just 25 million years, virtually every major animal group appeared simultaneously in the fossil record. We're discussing creatures with complex eyes, sophisticated nervous systems, intricate shells, and advanced hunting strategies. Trilobites emerged with compound eyes containing over 15,000 individual lenses, more sophisticated than most modern insects. Anomalocaris, a 6-foot predator, appeared with flexible arms and complex mouthparts designed for processing prey. Where are their ancestors? Where are the transitional forms showing gradual feature development? Traditional evolution predicts we should find millions of intermediate fossils showing step-by-step progression from simple to complex. Instead, we find gaps so vast that paleontologists name them the Cambrian Gap. The Cambrian explosion isn't unique. The fossil record contains similar sudden appearances. Birds appear in the Jurassic period without clear reptilian ancestors showing gradual flight development. Flowering plants emerge in the Cretaceous period without obvious precursors. Mammals diversify rapidly after dinosaur extinction, filling ecological niches with stunning speed. Even more puzzling, these rapid appearances often coincide with asteroid impacts and cosmic bombardment periods. The Permian-Triassic extinction, Earth's largest mass extinction, was followed by new life form explosions. The asteroid impact killing dinosaurs led to mammalian diversification. Each catastrophic event seems triggering biological innovation waves. Modern genetics adds another mystery layer. When scientists analyze DNA sequences across different species, they find many crucial genetic innovations appearing suddenly in the evolutionary timeline. Genes responsible for complex eyes, for instance, are remarkably similar across vastly different animal groups, suggesting they didn't evolve independently but were inherited from a common source. The Panspermia Hypothesis offers an alternative explanation. What if these sudden appearances represent biological colonization events? What if cosmic seeding doesn't just deliver simple organic compounds but actual genetic blueprints from civilizations that had already solved these evolutionary challenges? Between stars, in regions so cold atoms barely move, something extraordinary occurs. Vast cosmic dust clouds stretching across light years spontaneously create complex organic molecules. These aren't random chemical reactions. Radio telescopes have detected over 200 different organic compounds floating in interstellar space, including many essential life building blocks. The Eagle Nebula, 7,000 light-years away, contains enough organic material to seed millions of planets. The Orion Nebula produces alcohol molecules by the trillion along with sugars, amino acids, and more complex structure scientists are still identifying. These stellar nurseries aren't just forming new stars and planets, they're manufacturing biological chemistry on an industrial scale. How is this possible in space vacuum? The secret lies in tiny dust grains smaller than cigarette smoke particles. These microscopic factories provide surfaces where atoms can meet and bond, protected from harsh radiation that would normally tear molecules apart. Over millions of years, simple hydrogen and carbon atoms combine into complex structures. Think of each dust grain as a cosmic laboratory. Hydrogen atoms stick to surfaces and gradually accumulate. When ultraviolet light from nearby stars hits these grains, it provides just enough energy to drive chemical reactions without destroying products. The process resembles slow cooking on cosmic scales where patience and precise conditions create molecular complexity that shouldn't exist in deep space. James Webb Space Telescope observations have revealed something more astonishing. Some interstellar clouds contain molecular structures so complex they rival chemistry found inside living cells. Long-chain hydrocarbons, ringed organic compounds, and even primitive membrane structures have been detected floating in regions where temperatures are 400 degrees below zero. These discoveries fundamentally change how we think about life's building blocks distribution. Instead of organic chemistry being rare and confined to planetary surfaces, we now know it's happening continuously throughout the galaxy. Every star-forming region simultaneously churns out biological materials that get incorporated into new solar systems. On July 20th, 1976, something unprecedented occurred in human exploration history. The Viking 1 lander touched down on Mars's surface and immediately began conducting experiments designed detecting life signs. What happened next launched a scientific controversy continuing today and potentially provided first direct evidence of panspermia in action. The labeled release experiment was elegantly simple. The lander scooped Martian soil and mixed it with nutrient solution containing radioactive carbon. The logic was straightforward. If microorganisms existed in soil, they would consume nutrients and release radioactive carbon dioxide gas that sensitive detectors could measure. Results were immediate and dramatic. Within hours, detectors registered sharp spikes in radioactive gas production. The pattern looked exactly like what you'd expect from biological metabolism. Living organisms apparently eating nutrients and breathing out radioactive carbon dioxide. However, other experiments produced conflicting data. The gas chromatograph designed detecting organic molecules found no evidence of carbon-based compounds in Martian soil. How could living organisms exist without organic chemistry supporting them? For decades, scientific consensus dismissed Viking results as chemical rather than biological. Mars, they argued, was too cold, too dry, and too radiation-bombarded to support life. But this explanation never fully satisfied critics. Patterns detected by Viking were remarkably similar to biological signatures. Recent discoveries have forced scientists reconsidering these 40-year-old results. We now know Earth life exists in environments far more extreme than Mars's surface. Bacteria thrive in Antarctic ice, highly acidic volcanic springs, and environments with radiation levels lethal to humans. If life survives these conditions on Earth, why not on Mars? More importantly, we've learned that organic compounds can be destroyed by the same radiation that might allow life persisting underground. Viking landers only sampled surfaces, where organic molecules would be rapidly broken down by ultraviolet radiation and cosmic rays. Any organic chemistry supporting Martian life would likely exist meters below the surface, beyond Viking experiments' reach. If Viking experiments actually detected Martian life in 1976, it means we've had panspermia evidence for nearly 50 years. Mars and Earth, both targets of the same cosmic bombardment, both recipients of organic compounds from interstellar space, might host related life forms. Every living thing on Earth, from bacteria to blue whales, uses exactly the same genetic code. This universal language consists of just 4 chemical letters: A, T, G, and C. These letters combine into 3-letter words called codons, which tell cells how to build proteins. There are 64 possible 3-letter combinations, but they code for only 20 amino acids. This means the genetic code contains built-in redundancy, like a language designed surviving transmission errors. The mathematical precision is extraordinary. The genetic code is optimized to minimize mutation damage. If cosmic radiation changes 1 letter in a genetic word, redundancy ensures the resulting protein is usually still functional. But here's what makes scientists pause. This optimization level shouldn't exist if the genetic code evolved randomly on Earth. Computer simulations show millions of possible genetic codes would work perfectly fine for life on stable planets. Yet somehow, out of all these possibilities, Earth life uses the one code that happens to be nearly optimal for surviving cosmic radiation. The probability of this happening by chance is roughly 1 in a million. It resembles randomly generating a computer password and discovering it's perfectly optimized for security by someone understanding advanced cryptography. Recent genetic code analysis has revealed even stranger patterns. The way DNA letters are arranged follows mathematical principles that weren't discovered by human scientists until the 20th century. The genetic code shows evidence of error-correcting mechanisms, redundancy systems, and information compression techniques rivaling our most sophisticated computer programming. Some researchers have found what they call wow signals hidden in genetic sequences, mathematical patterns so precise and unlikely they appear intentionally embedded messages. The Fibonacci sequence, prime numbers, and other mathematical constants seem woven into DNA's fundamental structure itself. If these patterns are intentional, then the genetic code isn't just a random product of Earth's evolution. It's a design system created by intelligences understanding both biology and mathematics at levels we're only beginning appreciating. The timing adds another mystery layer. The universal genetic code appears established very early in Earth's history during the period when our planet was being heavily bombarded by asteroids and comets. Beneath 15 miles of solid ice, Europa hides an ocean twice as large as all Earth's seas combined. Jupiter's fourth largest moon shouldn't exist in any form supporting life. At 500 million miles from the Sun, surface temperatures plunge to minus 260 degrees Fahrenheit. Solar radiation is 25 times weaker than what reaches Earth. Yet something extraordinary happens beneath that frozen surface. Gravitational forces from Jupiter's massive pull flex Europa's rocky core, generating enough heat maintaining liquid water for billions of years. This subsurface ocean contains more water than all rivers, lakes, and seas on our planet. Recent analysis of Europa's thin atmosphere has detected water vapor plumes erupting from the surface, shooting hundreds of miles into space. These geysers provide a direct window into the hidden ocean below. When Hubble Space Telescope analyzed these plumes' chemical composition, it found something remarkable. The water signature matches Earth's oceans almost perfectly. The similarity isn't just chemical. Isotope ratios, acting like fingerprints for different water sources, suggest Europa's ocean and Earth's seas might share a common origin. This is difficult explaining if both worlds formed independently from local materials in their respective solar system regions. But Europa's connection to Earth might go far deeper than just water. The gravitational dynamics keeping Europa's ocean liquid also create powerful currents and chemical mixing that could support complex ecosystems. Volcanic vents on the ocean floor, similar to hydrothermal systems supporting life in Earth's deepest seas, might provide energy for biological processes. The most intriguing possibility is that Europa's ocean isn't just potentially habitable. It might be actively exporting life to other worlds. Every time those massive geysers erupt, they launch water, dissolved minerals, and potentially biological material into space. Some of this material escapes Europa's weak gravity and enters the broader solar system. Over billions of years, this process could have delivered countless biological packages from Europa's hidden biosphere to our planet's surface. The timing is perfect for early Earth during the late heavy bombardment period 4 billion years ago. 66 million years ago, a 6-mile-wide asteroid slammed into Earth with the force of 10 billion atomic bombs. The Chicxulub impact should have been the end of complex life on our planet. The explosion vaporized rock, created global tsunamis, and threw so much debris into the atmosphere that sunlight was blocked for months. 3/4 of all species went extinct, including dinosaurs that had dominated Earth for 160 million years. But something unexpected happened in this cosmic catastrophe's aftermath. Instead of a long, slow recovery, life exploded into forms more diverse and complex than ever before. Mammals, Previously small creatures living in dinosaurs' shadows, rapidly diversified into thousands of new species. Flowering plants flourished. New ecological niches were filled with stunning speed. This pattern isn't unique to the Chicxulub impact. Throughout Earth's history, major asteroid strikes have been followed by remarkable biological innovation. The correlation is so consistent that scientists have started mapping impact craters against the fossil record. What they found challenges conventional thinking about evolution and extinction. Rather than just destroying life, cosmic impacts seem introducing new biological possibilities. Consider what happens during a major impact. The explosion doesn't just kill organisms, it also creates conditions that could support cosmic hitchhikers' survival and growth. Massive heat and pressure can create hydrothermal systems persisting for thousands of years. These environments, similar to deep-sea vents where life might have first emerged on Earth, could provide perfect nurseries for extraterrestrial microorganisms. Recent computer simulations reveal that asteroid impacts can actually protect biological material during atmospheric entry. While surface rocks get heated to thousands of degrees, large meteorites' interiors can remain relatively cool. Microorganisms embedded deep within incoming asteroids could survive the journey and find themselves in environments specifically created by the impact event. This creates a perfect biological delivery system. Cosmic impacts simultaneously destroy existing ecosystems, creating evolutionary opportunities while delivering new genetic material that can exploit those opportunities. It's like clearing land and planting seeds in a single operation. Under a microscope, tardigrades look like tiny eight-legged bears wandering through a miniature landscape. These microscopic animals, smaller than salt grains, are found everywhere on Earth, in moss, lichen, soil, and water from Arctic to equator. But what makes tardigrades truly extraordinary isn't where they live, but what they survive. When conditions become hostile, tardigrades enter cryptobiosis, essentially becoming neither alive nor dead. Their metabolism drops to 0.01% of normal levels. They lose 99% of their body water and curl into an indestructible barrel called a ton. In this state, they survive conditions that would obliterate almost any other Earth life form. Temperature extremes that would instantly kill humans barely affect tardigrades. They've survived exposure to -458 degrees Fahrenheit, just degrees above absolute zero. They've also withstood temperatures of 300 degrees Fahrenheit. They can handle pressure 6 times greater than the deepest ocean trenches and radiation levels 6,000 times higher than what would be lethal to humans. Most remarkably, tardigrades are the only known animals surviving space vacuum. When European Space Agency researchers exposed tardigrades to harsh conditions outside the International Space Station for 10 days, 70% revived once returned to normal conditions. This resilience seems almost purposefully engineered for interplanetary travel. Tardigrades can remain in cryptobiosis for decades, potentially long enough to journey between nearby star systems. Their ability to repair DNA damage from radiation is more sophisticated than anything human technology has achieved. But here's what makes the tardigrade mystery even stranger. These survival abilities are completely unnecessary for their normal Earth lifestyle. Tardigrades typically live in mild-tempered environments where temperatures rarely exceed 100 degrees Fahrenheit and radiation levels are minimal. Why would evolution develop such extreme survival capabilities for organisms rarely encountering extreme conditions? Traditional evolutionary theory suggests traits develop in response to environmental pressures. Yet tardigrades seem equipped for environments far more hostile than anything they encounter on Earth. It resembles finding deep-sea diving equipment on animals living in shallow ponds. Recent genetic analysis has revealed another puzzling aspect of tardigrade biology. Their DNA contains genetic sequences that don't match any other Earth organisms. Roughly 20% of tardigrade genes appear acquired through horizontal gene transfer, but from unknown sources. Some scientists now suggest tardigrades might be living fossils from Earth's earliest biosphere, preserving survival traits from when our planet was regularly bombarded by asteroids and cosmic radiation. Others propose a more radical explanation. Tardigrades might be descendants of organisms that arrived on Earth from space, carrying genetic adaptations developed during interstellar journeys. Every 76 years, Halley's Comet returns to our inner solar system, carrying cargo more precious than any Earth treasure. As this ancient wanderer approaches the sun, solar radiation begins vaporizing its icy surface, creating the spectacular tale that has amazed humans for thousands of years. But hidden within that glowing display is something remarkable. Complex organic molecules, amino acids, and potentially even primitive life forms preserved in deep freeze for billions of years. Comets are time capsules from our solar system's earliest days. They formed in the Kuiper Belt and Oort cloud, regions so distant they preserve original materials from when planets first coalesced. Unlike asteroids heated and modified by their sun proximity, comets retain pristine samples of primordial chemistry. When the European Space Agency's Rosetta mission finally caught up with Comet 67P in 2014, discoveries revolutionized our understanding of these cosmic icebergs. The comet's nucleus contains 16 different amino acids, including glycine, the simplest protein building block. It harbored complex organic molecules like formamide, which plays crucial roles forming nucleotide bases making up DNA and RNA. But Rosetta found something even more extraordinary. The comet contained organic compounds organized into structures resembling primitive cell membranes. These weren't random chemical formations, but organized molecular arrangements that could potentially serve as containers for biological processes. The implications are staggering. If comets carry prebiological structures, they're not just delivering raw materials for life. They're shipping partially assembled biological kits that could accelerate evolution on any planet they encounter. Consider the delivery system scale. Our solar system contains trillions of comets in the Oort cloud alone. Throughout cosmic history, gravitational perturbations have sent these icy messengers careening toward inner planets. Earth has been bombarded by comets for over 4 billion years, each impact delivering tons of organic compounds directly to our planet's surface. The timing of major comet showers correlates remarkably with periods of rapid biological evolution on Earth. The late heavy bombardment 4 billion years ago coincided with first complex cells emergence. Periodic comet storms throughout geological history align with evolutionary leaps that paleontologists struggle explaining through gradual processes alone. In 1950, physicist Enrico Fermi asked a question that still haunts scientists. Where is everybody? The Fermi paradox highlights a troubling contradiction. Our galaxy contains over 400 billion stars, many with planetary systems that could support life. The universe is 13.8 billion years old, providing ample time for civilizations to arise and spread across stars. Yet despite decades of searching, we found no conclusive evidence of alien intelligence. Traditional explanations for this silence are deeply unsettling. Perhaps alien civilizations destroy themselves before achieving interstellar travel. Maybe intelligence is so rare we're truly alone in a vast empty cosmos. But the Panspermia Hypothesis suggests a radically different explanation. We're not looking for aliens because we are the aliens. Consider this possibility. Billions of years ago, when our galaxy was young and chaotic, advanced civilizations faced inevitable death of their star systems. Rather than accepting extinction, They developed the ultimate survival strategy, cosmic reproduction. They encoded their biological heritage into microscopic passengers that could survive interstellar journeys lasting millions of years. These ancient civilizations didn't just send random genetic material into space. They carefully designed self-replicating systems optimized for long-distance travel and rapid colonization of suitable worlds. The universal genetic code, with its sophisticated error correction and radiation resistance, wasn't the product of random evolution. It was engineered by intelligences understanding both biology and cosmic conditions at levels we're only beginning appreciating. From this perspective, Earth wasn't randomly chosen for seeding. It was deliberately prepared and monitored as part of a galaxy-spanning project to preserve and spread intelligence throughout the cosmos. The mathematical precision of our planet's orbit, the convenient presence of a large stabilizing moon, and the perfect atmospheric composition for complex life weren't cosmic accidents. They were results of careful planetary engineering. This scenario explains the Fermi paradox perfectly. We don't detect alien civilizations because they're not separate from us. We are their descendants, carrying their genetic legacy and slowly redeveloping their technological capabilities. Every human being is a walking monument to civilizations that existed billions of years before our solar system formed. In 1973, Francis Crick made a startling proposal that shocked the scientific community. Crick, who had won the Nobel Prize for discovering DNA structure, suggested that Earth life was too complex to have evolved naturally in the time available. Instead, he proposed that advanced extraterrestrial civilizations deliberately sent genetic material to our planet in unmanned spacecraft specifically designed to seed suitable worlds with life. This wasn't random speculation from a fringe theorist. Crick had spent decades studying cellular biology's intricate machinery and understood better than almost anyone how impossibly complex even the simplest living systems really are. His directed panspermia theory emerged from cold mathematical analysis of what evolution could and couldn't accomplish in Earth's brief history. The numbers that troubled Crick are even more troubling today. A single bacterial cell contains roughly 4 million precisely arranged molecular parts. The probability of these components spontaneously organizing into a functioning organism is so small mathematicians struggle expressing it. But directed panspermia solves this probability problem elegantly. Instead of requiring life to emerge from scratch on every planet, it suggests that intelligence, once achieved, takes responsibility for spreading itself throughout the galaxy. Advanced civilizations don't just survive by traveling to new worlds themselves. They ensure their biological legacy continues by deliberately seeding potentially habitable planets. Modern technology makes Crick's scenario increasingly plausible. We now understand how to design microscopic machines capable of self-replication and error correction. We know how to encode vast amounts of information in DNA sequences. We've identified genetic modifications that would allow organisms surviving extended space travel. If directed panspermia is real, then Earth's entire biosphere represents successful execution of an ancient terraforming project. Every forest, every ocean ecosystem, every creature that has ever lived carries the genetic legacy of civilizations that died billions of years before our sun ignited. 2.5 billion years ago, Earth's atmosphere underwent the most dramatic transformation in our planet's history. For over 1 billion years, our world had been shrouded in a toxic atmosphere of methane, ammonia, and carbon dioxide. Oxygen, the gas that now sustains complex life, was virtually non-existent. Then, in what geologists call the Great Oxygenation Event, everything changed. Oxygen levels skyrocketed from nearly 0 to 21% of the atmosphere in what amounts to a geological instant. This wasn't a gradual shift spanning hundreds of millions of years. It happened so rapidly it triggered the first mass extinction in Earth's history. Traditional science attributes this transformation to cyanobacteria, primitive organisms that developed photosynthesis and began converting carbon dioxide into oxygen. But the mathematics of this explanation creates serious problems. For cyanobacteria to produce observed atmospheric changes, they would need to multiply at rates exceeding anything observed in modern ecosystems, while simultaneously developing biochemical processes more sophisticated than most modern organisms possess. The speed of transformation is particularly puzzling. Computer models suggest natural photosynthesis should have taken over 10 billion years to create our current atmosphere. Yet, the Great Oxygenation Event accomplished this in less than 500 million years. But there's another explanation that makes the timeline work perfectly. What if the Great Oxygenation Event wasn't a natural evolutionary development but a deliberate terraforming operation? What if the cyanobacteria responsible for this atmospheric revolution were introduced as part of a cosmic seeding project specifically designed to prepare Earth for complex life? Evidence for this interpretation lies in photosynthesis's biochemical sophistication. This process requires two separate photosystems working in perfect coordination along with complex molecular machinery for capturing light energy and converting it into chemical energy. The probability of all these components evolving simultaneously by random chance approaches mathematical impossibility. The Perseverance rover is currently drilling into Martian rocks containing organic compounds identical to those found in Earth's earliest fossils. On February 18, 2021, this nuclear-powered laboratory landed in Jezero crater and immediately began finding evidence that Mars once hosted conditions suitable for life. But discoveries go far beyond just ancient water flows and suitable chemistry. Perseverance has detected complex organic molecules in rock samples showing characteristics of biological rather than geological formation. These aren't simple carbon compounds that could form through random chemical processes. They're sophisticated molecular structures that on Earth are produced exclusively by living organisms. Even more remarkable, some organic compounds show signs of recent formation. Martian soil samples contain molecules that appear to be actively changing, suggesting biological processes might still be operating in Mars's subsurface environment. Meanwhile, the James Webb Space Telescope has been detecting organic compounds in distant planets' atmospheres. These discoveries reveal that life's building blocks are common throughout the galaxy, not rare accidents confined to our solar system. The Europa Clipper mission, scheduled to arrive at Jupiter's moon in 2030, will search for life signs in the subsurface ocean containing more water than all Earth's seas combined. Preliminary analysis of water vapor plumes erupting from Europa's surface has already detected organic compounds and possible amino acids. Perhaps the most intriguing discoveries are coming from Earth's own upper atmosphere. High-altitude research flights have detected microorganisms living in the stratosphere 25 miles above the surface where conditions approach those of space itself. These organisms seem specifically adapted for surviving in near-vacuum conditions with intense radiation exposure. Some high-altitude organisms don't match any known Earth species. Their genetic signatures suggest they might be recent arrivals from space rather than terrestrial life forms that drifted upward. If microorganisms are regularly arriving from space, they could be continuously introducing new genetic material into Earth's biosphere. Evolution has a speed limit, and Earth life constantly breaks it. Charles Darwin understood that natural selection requires vast amounts of time creating complex biological systems. Each beneficial mutation must spread through populations, accumulate with other favorable changes, and gradually build the intricate molecular machinery powering living cells. The mathematics of this process predicts major evolutionary innovations should take hundreds of millions of years developing. Yet Earth's fossil record tells a completely different story. Complex biological systems appear suddenly, function perfectly from their first appearance, and show no evidence of the gradual assembly Darwinian evolution requires. The numbers are staggering. Developing a single new protein through random mutation and natural selection should take roughly 100 million years under optimal conditions. Yet, the Cambrian explosion introduced hundreds of new protein families simultaneously in just 25 million years. Consider the complexity involved. A bacterial flagellum, the rotating motor propelling many bacteria, contains over 40 different protein components that must work together with clockwork precision. Remove any single component and the entire system fails. Evolutionary biologist Michael B. calculated that assembling such a system through random mutations would require more time than the universe has existed. But panspermia solves the timescale problem elegantly. Instead of requiring each planet to independently evolve complex biological systems from scratch, cosmic seeding allows worlds to inherit pre-designed genetic modules that were developed over billions of years by civilizations throughout the galaxy. From this perspective, Earth's rapid biological innovations make perfect sense. When cosmic seeding packages arrive on our planet, they don't just deliver simple organic compounds. They bring complete genetic blueprints for sophisticated biological machines that were perfected on countless other worlds. The sudden appearance of complex systems in Earth's fossil record reflects the arrival and activation of these cosmic genetic libraries. Recent research in synthetic biology supports this interpretation. When human scientists try designing biological systems from scratch, they invariably create modular architectures where complex functions are built from interchangeable genetic components. This is exactly the pattern we see in Earth's most sophisticated biological systems. The genetic modules responsible for complex traits like photosynthesis, nervous systems, and immune responses show characteristics of engineered rather than evolved systems. They're too well integrated, too mathematically optimized, and too functionally sophisticated to be products of random mutation and natural selection operating within Earth's limited time frame. On July 25, 2001, the skies over Kerala, India began raining blood. For 2 months, crimson droplets fell from clear skies across a 100-square-mile area. Local residents reported that the rain left red stains on clothing and buildings that were nearly impossible to remove. Government officials initially dismissed the phenomenon as dust from distant deserts. But laboratory analysis revealed something far more extraordinary. The red coloration came from microscopic cells unlike anything in Earth's known biology. These organisms were roughly 4 micrometers in diameter, about half the size of typical bacteria, with thick cell walls and unusual internal structures. When scientists tried culturing them using standard biological techniques, the cells refused to grow in any known nutrient medium. But here's where the story becomes truly remarkable. The red rain cells showed characteristics that seemed specifically adapted for surviving in space. They could withstand extreme temperatures, intense radiation, and complete dehydration. Dr. Godfrey Lewis, a physicist at Mahatma Gandhi University, spent years analyzing these cells and reached a startling conclusion. The organisms weren't terrestrial. Their cellular structure, chemical composition, and survival characteristics all suggested an extraterrestrial origin. Most significantly, they contained no DNA or RNA, the genetic molecules that define all known Earth life. Instead of conventional genetic material, the red rain cells appeared to use an entirely different information storage system. They reproduced by a process resembling budding more than cellular division, and they could apparently survive indefinitely without nutrients or water. The timing of the red rain events added another mystery layer. Many incidents occurred shortly after meteor showers or unusual atmospheric phenomena. Witnesses reported seeing bright flashes in the sky before the red droplets began falling. Some scientists suggested the cells were being delivered by micrometeorites too small to be detected by conventional monitoring systems. Kerala wasn't an isolated case. Similar red rain events have been reported throughout history and around the world. Ancient texts describe blood rain falling during significant historical events. Modern incidents in Colombia, Austria, and Sri Lanka have yielded microscopic organisms with characteristics remarkably similar to the Kerala cells. Chemical analysis of these mysterious organisms has revealed isotope ratios that don't match terrestrial biology. The carbon signatures in particular suggest formation in environments with different stellar conditions than our solar system. These aren't just unusual Earth organisms. They appear to be genuine alien life forms. Imagine playing cosmic roulette where the stakes are intelligence's survival itself across the entire galaxy. Traditional approaches to interstellar colonization require massive resources and enormous risks. Building starships capable of carrying living passengers between star systems demands technologies and energy sources that stretch even advanced civilizations to their limits. Journey times span thousands of years during which equipment fails, resources deplete, and colonists face psychological pressures that could destroy entire missions. But panspermia changes the mathematical equation completely. Instead of sending a few expensive, fragile spacecraft to carefully chosen destinations, civilizations can launch billions of microscopic survival packages toward every potentially suitable planet in their galactic neighborhood. The cost per mission drops to nearly zero while the probability of success skyrockets. Consider the numbers. Our galaxy contains over 400 billion stars, many with planetary systems that could support life. A single dying civilization could potentially seed thousands of nearby star systems using technology no more advanced than our current genetic engineering capabilities. Each seeding package would be designed to survive interstellar journeys lasting millions of years while carrying complete biological blueprints for reconstructing complex life. The mathematics of survival probability become overwhelming in favor of the seeding strategy. If a civilization sends just 100 seeding packages to each of 1,000 nearby star systems, they've launched 100,000 biological time capsules into space. Even if 99% of these missions fail due to radiation damage, impact destruction, or unsuitable planetary conditions, 1,000 seeding operations still succeed. Each successful seeding event doesn't just create life on a new world. It establishes a biological beachhead that can eventually redevelop intelligence and technology. Given enough time, these new civilizations reach the point where they can launch their own seeding missions, creating an exponential expansion of intelligence throughout the galaxy. The timescales involved work in favor of this strategy. While conventional space travel requires civilizations to remain stable for thousands of years during transit, panspermia operates on geological timeframes where even unstable civilizations can achieve galactic impact. Recent computer simulations of galactic panspermia show that even conservative seeding rates could populate the entire Milky Way with life within 2 billion years. Given that our galaxy is over 13 billion years old, there's been more than enough time for multiple waves of cosmic seeding to occur. The redundancy built into this system makes extinction nearly impossible. Unlike conventional colonization where the destruction of individual colonies threatens the entire expansion effort, panspermia creates thousands of independent backup copies of civilization. On August 15, 1977, astronomer Jerry Emin detected a radio signal so powerful and precisely focused that he wrote, "Wow," in the margin of the computer printout. The signal lasted exactly 72 seconds and came from the direction of the constellation Sagittarius. Its frequency was 1,420 megahertz, precisely the wavelength that hydrogen atoms emit when they change energy states. This wasn't random cosmic noise, It was a narrowband transmission requiring technological sophistication to generate. Despite decades of searching, the WOW signal has never been detected again. Traditional SETI researchers assume it was a one-time transmission from an alien civilization. But the Panspermia Hypothesis suggests a radically different interpretation. What if the WOW signal wasn't a technological transmission at all but a biological one? What if it was the electromagnetic signature of a massive cosmic seeding event where billions of genetic packages were being launched toward our solar system? Recent research has revealed that biological processes can generate electromagnetic emissions under specific conditions. When DNA replicates, it produces measurable electromagnetic fields. When cells divide rapidly, they create electromagnetic pulses that can be detected with sensitive equipment. Large-scale biological events involving trillions of organisms simultaneously could potentially generate radio signals powerful enough to be detected across interstellar distances. If ancient civilizations encoded genetic information into electromagnetic patterns, they could transmit biological blueprints at the speed of light rather than waiting millions of years for physical delivery. These transmissions wouldn't be trying to establish communication in the traditional sense. They would be broadcasting genetic instructions that could be received and implemented by any civilization with sufficient technological capability. The mathematics of this approach are compelling. A single electromagnetic transmission lasting just minutes could carry more genetic information than billions of physical seeding packages. The signal could be precisely targeted at star systems showing signs of developing intelligence, allowing advanced civilizations to accelerate the biological development of younger worlds. Deep beneath the Australian outback lies a rock formation that shouldn't exist on any planet formed through natural processes. The Pilbara Craton contains mineral structures dating back 3.5 billion years that show characteristics typically associated with biological activity. These aren't simple organic compounds, but complex crystalline formations that appear to have been shaped by living organisms using techniques that exceed anything in modern biology. Geologists studying these ancient formations have found microscopic tubes, spheres, and filaments embedded in rock that formed when Earth was still a hostile, radiation-bombarded world. The precision of these structures suggests they were created by organisms with sophisticated tools for manipulating matter at the molecular level. Similar anomalous formations have been discovered in geological sites around the world. The Barberton Greenstone Belt in South Africa contains mineral deposits with isotope ratios that don't match any known terrestrial processes. The Isua supercrustal belt in Greenland shows evidence of biological carbon fixation occurring at temperatures and pressures that should have prevented any known form of life from surviving. But these ancient biological signatures aren't limited to obvious fossil sites. Some of Earth's most fundamental geological features show characteristics suggesting artificial modification rather than natural formation. The distribution of rare Earth elements in certain rock layers follows patterns that are mathematically too precise to be random. Recent analysis of impact crater formations has revealed another puzzling pattern. Many ancient impact sites show unusual mineral compositions, suggesting the incoming objects weren't typical asteroids, but contained materials with biological origins. Chemical signatures in these craters indicate the presence of complex organic compounds that formed under conditions unlike anything in our solar system. The Sudbury impact structure in Canada, formed 1.85 billion years ago, contains mineral deposits showing evidence of rapid biological colonization immediately after the impact event. The speed of this colonization exceeds anything observed in modern impact sites, suggesting the incoming object delivered organisms specifically adapted for exploiting post-impact environments. Even more intriguing are geological formations that appear to have been deliberately constructed rather than naturally formed. The Rikhat structure in Mauritania, often called the Eye of the Sahara, shows concentric ring patterns with mathematical precision that's difficult to explain through conventional geological processes. Underground cave systems in various parts of the world contain mineral formations that seem to have been shaped by unknown biological processes. These structures show levels of organization and complexity exceeding what modern organisms can achieve. The caves themselves often follow geometric patterns, suggesting purposeful excavation rather than natural water erosion. Life on Earth has endured countless catastrophes. Asteroid impacts, supervolcanic eruptions, global ice ages, and radiation bursts. Each event reshaped evolution's trajectory. Yet through all this, life persisted. That persistence implies not just resilience but a deep continuity that binds all organisms together. If panspermia is true, then this continuity stretches beyond our planet. The same molecular patterns, DNA, amino acids, or their precursors, may link us to distant worlds we have never seen. In that sense, Earth itself could be one node in a much larger biological network spanning light years. Humanity's emergence would then represent the universe becoming self-aware through one of its many branches of life. However, continuity does not guarantee permanence. Every civilization, no matter how advanced, remains vulnerable to both internal and cosmic forces. Climate change, resource depletion, asteroid impacts, or self-inflicted collapse. The absence of visible galactic civilizations, despite the high probability of habitable worlds, is what the physicist Enrico Fermi called the Great Filter. Something, perhaps many things, limits the survival of intelligent species. If we are indeed part of a chain of cosmic life, our challenge is to ensure that the chain does not end with us. Modern research in astrobiology and planetary science has begun to identify potential bridges through which life could travel between worlds. Mars, for instance, was once warmer and wetter than it is today. Meteorites ejected from its surface by asteroid impacts have been found on Earth, proving that natural exchange of material between planets in the same system is possible. If microorganisms existed on early Mars, some might have hitched a ride to Earth or vice versa. Similarly, icy moons such as Europa, Enceladus, and Titan harbor subsurface oceans and complex chemistry. Impacts on these worlds could eject frozen material into space, potentially seeding other bodies. Even interstellar objects like Oumuamua and Borisov hint at the possibility of matter, and perhaps microscopic life drifting between star systems. Whether through random exchange or directed effort, these pathways blur the boundary between planetary and cosmic biology. They invite us to imagine life as a phenomenon that can spread, adapt, and perhaps communicate across astronomical distances. From a philosophical perspective, the panspermia framework reshapes how we interpret human consciousness. If life is universal and intelligence emerges repeatedly given enough time, then awareness itself might be an inevitable feature of cosmic evolution. In this view, the universe is not a lifeless void occasionally sprinkled with biology, but a living, creative system seeking to know itself. Humanity's curiosity, the drive to explore, to build telescopes, to ask questions, is a natural extension of that process. We are the cosmos reflecting upon its own existence. This idea echoes the thoughts of thinkers like Carl Sagan, who wrote that we are a way for the cosmos to know itself, and of modern cosmologists who speak of the anthropic principle, that consciousness and the universe are intertwined in ways still poorly understood. Whether framed scientifically or poetically, the implication remains that life and mind are not alien intrusions into a mechanical cosmos. They are the universe's own self-organizing possibilities made real. Recognizing that we may be part of a lineage of cosmic life carries ethical consequences. If living matter is widespread, then planets like Earth are not merely resources but sacred archives of evolution. To destroy ecosystems or drive species to extinction is to erase chapters of a universal story billions of years in the making. Similarly, if other worlds host life, even microbial, our future explorations must proceed with humility and care. The field of planetary protection already reflects this awareness, enforcing sterilization protocols for spacecraft to prevent biological contamination of other planets. In the long run, this ethic may need to expand beyond science to guide how humanity interacts with the broader cosmic environment. At the same time, acknowledging our potential cosmic kinship could foster a renewed sense of unity among humans themselves. We are not separate nations or tribes competing for survival, but a single species carrying the memory of the stars. Our divisions pale beside the vastness of time and space from which we arose. Returning to the question of earlier civilizations, whether terrestrial or extraterrestrial, it is possible that the universe is filled not only with life but also with ruins. Civilizations may rise and fall across eons, leaving traces too faint for us to detect. Dyson spheres, derelict satellites, or molecular fossils might drift unseen in interstellar darkness. Even on Earth, if another intelligent species once flourished, its legacy might now lie deep beneath the ocean floor or buried under layers of basalt. Perhaps what we call the myths of the gods or lost ages in ancient cultures echo distant memories of past epochs. Although such ideas remain speculative, what matters more is the insight these questions provide. Civilizations are temporary, but life endures. If we are not the first, then we are the latest stewards of a cosmic heritage, inheritors of a long experiment that began before Earth was born. If we are the first, then we carry the extraordinary responsibility of ensuring that life's journey continues beyond us. Ironically, the very concept of panspermia might soon move from theory to practice. With humanity's increasing ability to send spacecraft beyond the solar system, the possibility of intentional panspermia, the deliberate seeding of life, enters ethical and philosophical debate. Should we one day send microorganisms to lifeless worlds to ensure the continuation of biology beyond Earth? Or should we preserve those worlds in their pristine natural state? Either choice would make us participants, not merely observers, in the cosmic cycle of creation. It would mark a transition from a species confined to one planet to a caretaker of life on a galactic scale. Our technologies, for all their risks, could also serve as the means by which life expands its domain and safeguards its future against extinction. In this way, panspermia might not only describe our past but also define our destiny. Life on Earth has endured countless catastrophes. Asteroid impacts, supervolcanic eruptions, global ice ages, and radiation bursts. Each event reshaped evolution's trajectory. Yet through all this, life persisted. That persistence implies not just resilience but a deep continuity that binds all organisms together. If panspermia is true, then this continuity stretches beyond our planet. The same molecular patterns, DNA, amino acids, or their precursors, may link us to distant worlds we have never seen. In that sense, Earth itself could be one node in a much larger biological network spanning light years. Humanity's emergence would then represent the universe becoming self-aware through one of its many branches of life. However, continuity does not guarantee permanence. Every civilization, no matter how advanced, remains vulnerable to both internal and cosmic forces, climate change, resource depletion, asteroid impacts, or self-inflicted collapse. The absence of visible galactic civilizations, despite the high probability of habitable worlds, is what the physicist Enrico Fermi called the Great Filter. Something, perhaps many things, limits the survival of intelligent species. If we are indeed part of a chain of cosmic life, our challenge is to ensure that the chain does not end with us. Modern research in astrobiology and planetary science has begun to identify potential bridges through which life could travel between worlds. Mars, for instance, was once warmer and wetter than it is today. Meteorites ejected from its surface by asteroid impacts have been found on Earth, proving that natural exchange of material between planets in the same system is possible. If microorganisms existed on early Mars, some might have hitched a ride to Earth or vice versa. Similarly, icy moons such as Europa, Enceladus, and Titan harbor subsurface oceans and complex chemistry. Impacts on these worlds could eject frozen material into space, potentially seeding other bodies. Even interstellar objects like Oumuamua and Borisov hint at the possibility of matter, and perhaps microscopic life drifting between star systems. Whether through random exchange or directed effort, these pathways blur the boundary between planetary and cosmic biology. They invite us to imagine life as a phenomenon that can spread, adapt, and perhaps communicate across astronomical distances. From a philosophical perspective, the Panspermia framework reshapes how we interpret human consciousness. If life is universal and intelligence emerges repeatedly given enough time, then awareness itself might be an inevitable feature of cosmic evolution. In this view, the universe is not a lifeless void occasionally sprinkled with biology, but a living, creative system seeking to know itself. Humanity's curiosity, the drive to explore, to build telescopes, to ask questions, is a natural extension of that process. We are the cosmos reflecting upon its own existence. This idea echoes the thoughts of thinkers like Carl Sagan, who wrote that we are a way for the cosmos to know itself, and of modern cosmologists who speak of the anthropic principle, that consciousness and the universe are intertwined in ways still poorly understood. Whether framed scientifically or poetically, the implication remains that life and mind are not alien intrusions into a mechanical cosmos. They are the universe's own self-organizing possibilities made real. Recognizing that we may be part of a lineage of cosmic life carries ethical consequences. If living matter is widespread, then planets like Earth are not merely resources but sacred archives of evolution. To destroy ecosystems or drive species to extinction is to erase chapters of a universal story billions of years in the making. Similarly, if other worlds host life, even microbial, our future explorations must proceed with humility and care. The field of planetary protection already reflects this awareness, enforcing sterilization protocols for spacecraft to prevent biological contamination of other planets. In the long run, this ethic may need to expand beyond science to guide how humanity interacts with the broader cosmic environment. At the same time, acknowledging our potential cosmic kinship could foster a renewed sense of unity among humans themselves. We are not separate nations or tribes competing for survival, but a single species carrying the memory of the stars. Our divisions pale beside the vastness of time and space from which we arose. Returning to the question of earlier civilizations, whether terrestrial or extraterrestrial, it is possible that the universe is filled not only with life but also with ruins. Civilizations may rise and fall across eons, leaving traces too faint for us to detect. Dyson spheres, derelict satellites, or molecular fossils might drift unseen in interstellar darkness. Even on Earth, if another intelligent species once flourished, its legacy might now lie deep beneath the ocean floor or buried under layers of basalt. Perhaps what we call the myths of the gods or lost ages in ancient cultures echo distant memories of past epochs, although such ideas remain speculative. What matters more is the insight these questions provide. Civilizations are temporary, but life endures. If we are not the first, then we are the latest stewards of a cosmic heritage, inheritors of a long experiment that began before Earth was born. If we are the first, then we carry the extraordinary responsibility of ensuring that life's journey continues beyond us. Ironically, the very concept of panspermia might soon move from theory to practice. With humanity's increasing ability to send spacecraft beyond the solar system, the possibility of intentional panspermia, the deliberate seeding of life, enters ethical and philosophical debate. Should we one day send microorganisms to lifeless worlds to ensure the continuation of biology beyond Earth? Or should we preserve those worlds in their pristine natural state? Either choice would make us participants, not merely observers, in the cosmic cycle of creation. It would mark a transition from a species confined to one planet to a caretaker of life on a galactic scale. Our technologies, for all their risks, could also serve as the means by which life expands its domain and safeguards its future against extinction. In this way, panspermia might not only describe our past but also define our destiny. All these perspectives converge on a single idea. Humanity is part of a much larger story, one that began long before us and will continue long after. We are the children of stardust, the descendants of molecules forged in ancient supernovae, arranged by chance and chemistry into the fragile complexity of life. Whether life first sparked on Earth or arrived from distant worlds, the result is the same. We are the universe achieving self-awareness. To speak of humanity's cosmic heritage is to recognize that our lineage is not limited to our planet or our species. It encompasses the entire chain of matter and energy that gave rise to consciousness. Every atom in our bodies once belonged to stars. Every breath we take connects us to an atmosphere shaped by billions of years of biological activity. Every act of curiosity and compassion reaffirms the universe's capacity to create meaning within itself. Understanding this heritage can transform how we see both science and ourselves. Panspermia is not merely a hypothesis about microbial exchange. It is a reminder that life's boundaries are wider than we once imagined. The question of whether we are the first civilization becomes less about pride and more about participation. We are part of an unfolding experiment in complexity, creativity, and awareness. Our task, then, is to honor and extend that experiment, to explore without arrogance, to innovate without destruction, and to preserve the delicate chain of life that connects us to the cosmos. Whether we are alone or one among many civilizations, our existence affirms that the universe is capable of knowing itself through us. Humanity's cosmic heritage is both a gift and a responsibility. It invites us to look beyond our divisions, beyond our short histories, and see ourselves as part of something ancient and ongoing, a living universe still writing its story through us. In the end, the Panspermia hypothesis may or may not prove scientifically true in every detail. But even as a framework of imagination, it reminds us that life is not an isolated miracle, but a universal melody. We are notes in that vast composition, resonating across time and space. The atoms that once drifted between stars now think, dream, and ask questions. And perhaps somewhere far away, other minds are doing the same, wondering, as we do, how their story began and whether they, too, are the first.